Hi, everybody. Good morning. We're trying to start off on time because we want to set a precedent. Sorry, we're 2 minutes late. This week is going to be jam-packed, and you're going to be running between here, Grand Ballroom Salon A, and downstairs, and back up here, and we hope you have a wonderful time. I'm Mo Plenary. I'm the Senior Collections Manager in Ornithology and Mammalogy at the California Academy of Sciences.
And I'm Laura Eklund, the Senior Collection Manager of Anthropology at the California Academy of Sciences. And Mo and I, as many of you are probably aware because you've gotten tons of emails from us, we are the co-chairs for the SPNHC 2023 Local Organizing Committee.
Oh, good. Okay. I was looking up. So we want to welcome you all in person and live streamed here. We are going out into the world in YouTube. Hopefully it's all working. So hello everyone out in YouTube. We just want to thank you all for coming. This session has probably 420 people in it today, which is one of the largest in-person SPNHC meetings. And I just want to go through a little bit before we start. This is the 38th annual meeting. How many of you have been at a SPNHC meeting since 38 years ago? Anybody? Yeah, raise your hand. 38 years ago? We've got a few handfuls. Thank you for being a committed member to the organization for all that time. How many people like me in the last 20 years have been to 10 or 12 meetings, something like that? Okay, good number. I'm in there with you too. Now, how many people is this your first meeting? Look at that. Over half of the attendees this year in person are first-time attendees, and we could not be happier about that. There's an emerging professionals lunch tomorrow. You'll all get to meet each other, but just take a look around and get to know each other, new and old.
So we are, of course, meeting here today in person, for those of us in person, in San Francisco, California, at the Hilton Union Square with our host institution, the California Academy of Sciences. And I would just like to take a moment to recognize that we are standing on Ohlone land, and I want to honor and appreciate all of the ancestors and the elders and the next generation of Ohlone people for all that they have done, and I honor that. Thank you.
Moving on, another note, we also want to thank our sponsors. You will see all of them here on the slide throughout the conference. We're very grateful to them for making this conference possible. Please interact with them in Grand Ballroom Salon B, which is also where you'll be getting your food, all your breaks, your lunch, and that's where you can really interact with our wonderful sponsors. So our two diamond sponsors are Picture A and Delta Designs Limited. Delta has been a long time sponsor of SPNHC meetings, and we're so happy to have them back this year. They have wonderful products, and they're great folks to talk to. Please spend time with them. Picture A is very new to us. We're so happy to have them here at SPNHC. Some of you might have met them yesterday during the workshop because they're actually working on a project at the Academy. They will have an open house on Friday that people can sign up for if you're free on Friday afternoon. And they will also be in the exhibit hall. So please spend time talking to the wonderful people from Picture A. If you want to visit our sponsors elsewhere than the exhibit hall, you can visit them through the virtual app I'm sorry, it is not an app. I was not going to say the word app, and I said the word app. It is a platform. Please visit the virtual booths in the Oxford Abstracts platform. You can also interact with other delegates through the Oxford Abstracts platform. So make use of it. The program is all in there. And if you have any problems, just ask anyone with a orange event staff badge. They should be able to help you.
And the sponsors have green badges.
Oh, and the sponsors have green badges.
Wait, one more thing.
Sorry. No, this is going to be the story of the week, right? Get Mo off the stage. Anyway, this session, the plenary session, is especially sponsored by GBIF and iDigBio as our gold sponsors. So they also will be in the exhibit hall and also on the virtual booths. They have workshops and sessions as well. So please visit the folks from GBIF and iDigBio, and we're very grateful to them too.
And all of our wonderful sponsors have in-person and virtual presence, and we really could not have put this conference on without them. Huge thanks to all of them, and please do take the time to visit with them and learn about their great products. So a SPNHC conference traditionally begins with a welcome address from the director of the host institution. And as it turns out, the director of the academy, Dr. Scott Sampson, is actually in Paris right now, meeting with the leaders, the directors of a number of other natural history museums. We cannot make this up. True story. So he's unable to be with us in person. However, he did record a welcome message, which we would like to play for you now.
It's an understatement to say that the Academy is very pleased to host SPNHC 2023. I'd like to begin by acknowledging that our museum sits on the ancestral territory of the Ramaytush Ohlone. The Ohlone nations never ceded their territory, nor did they set aside their responsibilities to be in a reciprocal relationship with this land. We honor the sovereignty, ecological knowledge, and caretaking practices of Indigenous nations in California and beyond, knowing that we have much to learn from Native peoples. We acknowledge the responsibility held by institutions like ours for the erasure and exclusion of Indigenous peoples, and today, we strive for justice. The main theme of this year's conference is taking the long view. As a paleontologist, taking the long view comes pretty naturally, but most of us today tend to suffer from what I like to call severe temporal myopia. Time is something that extends at most between grandparents and grandchildren. We struggle to imagine life 100 years ago, let alone 20,000. We fail to see the countless interwoven ecological threads all around us, forged over millennia. And we're wedded to the notion of stasis, envisioning our home places and all other places persisting virtually unchanged through time. Does this limited sense of time really matter? In a word, yes. Indeed, it has the potential to be deadly on a grand scale. We are all players in a long-running eco-evolutionary drama, but we've forgotten our roles. We fail to see the dynamism of the world, the way our local ecosystems continually adapt and evolve as conditions shift. So we're left unable to respond to the needs of our moment. To make matters worse, human-driven climate warming, habitat destruction, and biodiversity losses have greatly accelerated the pace of transformation. In short, we desperately need to embrace the concept of change, envisioning ways we can work with the natural world rather than fighting against it. What does all this have to do with natural history museums? Well, collectively, we are the keepers of Earth's story. We are holders of the objects that demonstrate how Earth and life have changed through time. This year's SPNHC theme encourages us to draw lessons from the past to shape a better future for our field and our collections. But I humbly suggest that as you think about taking the long view, you consider a much bolder aim. I believe we have the potential to envision a future in which natural history museums use their collections and knowledge of the past to shape a flourishing future for interwoven communities of humans and non-humans alike. Here at the California Academy of Sciences, our mission is to regenerate the natural world through science, learning, and collaboration. I probably don't have to tell this group that natural history collections as libraries of life hold important lessons not only for understanding the past, but also for regenerating ecosystems on a warming world. Yet, because of our field's history of extraction, a history we must now reckon with, these collections still are not as widely accessible as they should be. Today, we are collectively taking important steps to address this inequity through efforts to democratize our collections, through digitization, as well as through the Global Natural History Initiative announced in March. At the Academy, we're also training young scientists in other nations on the front lines of climate change, particularly island nations, to become conservation leaders in their own home countries. In the Maldives, we're supporting leaders as they work to build their own natural history museum and collections. These are just a few of the examples of what's possible. We can and should do more. The conference theme also asks, what are our goals as a society and as individual organizations? Science-based cultural organizations, including natural history museums, are in a unique position to shift the now globally pervasive feeling of doom and despair. Our assets include high public trust, amazing venues to host exhibits, programs, convenings, and of course, our collections and the deep expertise in science that is required to preserve them. While fully acknowledging the dire nature of this pivotal moment, we can help spread a more positive, empowering story of humanity's role in the world. A story in which people align with the rest of the natural world to regenerate the health of social and ecological communities. A story with potential to help grow the intersectional movement needed to tackle social justice, climate, and the biodiversity crisis. Imagine if we all combined forces to help build this movement to inspire millions of people around the world to understand, care about, and act on behalf of nature. So, as you meet to discuss the future of collections, I invite you to consider ways that we could leverage our collections and each other to shape and scale solutions, as well as to share success stories. We look forward to showing you a sampling of our 46 million objects during our collection tours. And we hope you find some time just to walk around our public floor and enjoy Golden Gate Park. Thank you so much for attending SPNHC 2023 here in San Francisco, and a big thank you to our sponsors for your generous support. I have no doubt that you'll have a fantastic time this week.
Thank you, Dr. Sampson. Maybe he's out there in cyberspace listening to us on YouTube, like so many of our colleagues are. So we now have 3 in-person plenary speakers, and we know you're really going to enjoy all of their talks and getting to know them throughout the week. And in order to introduce them, I would like to introduce Mo and my boss, the very long title. So I have the Harry and Diana Hind, Dean of Science and Research Collections, Dr. Shannon Bennett.
Thank you, Mo and Laura, and thank you everybody for being here. I can't tell you how excited I am to see you all in person and to host this august group of people in the SPNHC meeting here in San Francisco and at the California Academy of Sciences. You're going to have a great time tonight at the Icebreaker at CAS, so looking forward to it. So it gives me great pleasure to introduce our first plenary speaker, Dr. Luiz Rocha. Luis is the Follett Chair and Curator of Ichthyology at the California Academy of Sciences. He's also co-director of the Hope for Reefs Initiative, one of Cal Academy's strategic initiatives. And the title of his talk, first of all, I'll get to that in a second. So I want to just say a few words about Luis. His major research interests include evolution, conservation, taxonomy, and community ecology of reef fishes. Coral reef fishes. He has spent over 6,000 hours underwater studying these fishes and published over 170 articles and 2 books. He's also been featured in popular media For example, CNN, Scientific American, the New York Times, the Science Channel, and National Geographic. And most importantly, his work has supported conservation efforts around the globe. His current main focus of work is exploring little-known deep reefs between 200 and 500 feet. These are reefs that are so unexplored as ecosystems that they remain largely unprotected. And so his current passion is to describe the uniqueness of these fauna in these depths and advocate for strategic conservation, either expansion of existing marine protected areas or new deep reef focused marine protected areas. And the creation of these conservation areas and this focus was recently honored in 2021 by a Rolex Award for Enterprise. So his talk is Ecology, Taxonomy, and Conservation of Fishes in Mesophotic Coral Ecosystems. Please join me in welcoming Dr. Rocha.
Thanks, Shannon. Good to be here. Welcome, everybody, to nice San Francisco. I hope you have a good time here. I will start by saying that I think 6 or 7 different people told me do not hit the escape button. And then there's a big green tape here on top of the escape button that says do not press it. Apparently, if I press it, the live stream stops. So if the live stream stops, you know why it stopped, because my hand gets like dangerously close to that, and I'm tempted just to see what happens. Anyhow, deep reefs, that is what I've been doing here since I joined the Academy. It's a very complex, as you see as we go through the talk, it's a very complicated work to do because of the type of diving that we do. So it requires a special kind of institution. It's very hard to do this on a university, for example, because of the bureaucracy and their general aversion to risk. So doing this at a museum, at a natural history museum, it's much easier because people like the risk factor, and it's easier it's flashier to attract donors to fund this kind of stuff on a natural history museum as opposed to a university. NSF doesn't want anything to do with this because they think it's too risky, both from a diving perspective, but also from a scientific perspective, because as Shannon said, we know almost nothing about those deep reefs. But before I go deep, I want to talk a little bit about shallow reefs, just to make sure that everybody is on the same page here. Shallow reefs, they are distributed in a very narrow area in the ocean. They require very shallow water, they require light, and they require light because the corals, the ones that build the ecosystem, they live in symbiosis with this alga, it's called the zooxanthellae. So the zooxanthellae lives within the coral tissues, a very close, tight relationship, and one survives through the nutrients that are expelled by the other. So it's a very nice recycling system that allows these ecosystems to prosper in otherwise very waters that are very poor in nutrients. So the clear water means there's very few nutrients in it, and the nutrients they get from the algae, and then the algae get the nutrients from the coral. So because of this relationship, then this very diverse ecosystem developed. And by very diverse, I mean in the ocean, there's nothing that even comes close. This diversity is very disproportionately distributed in the ocean. So coral reefs, they cover less than 0.1% of the surface of the ocean, but about 25% of all marine species live only on coral reefs. And that's for all taxa. So 25% of all marine fish only in coral reefs, 25% of all crustaceans only in coral reefs. And the area is less than 0.1%. So one of the biggest drivers around my whole research line is trying to understand why there are so many species on reefs and how they can coexist. They can coexist together. But I'm sure that as people that are interested in natural history and interested in the natural world in general, you probably heard about risks that are and impacts that are affecting coral reefs lately. Those risks go from local to global. They range from everything from overfishing to plastic pollution to like the physical destruction of reefs to sewage pollution that increases the nutrient in the water and then favors the algae that ends up overgrowing the coral. So lots of local threats. But today, the biggest threat is more global in nature. So this is a trip, an expedition that we did with the Academy team to French Polynesia. This was in April 2019. We went there, we spent 2 weeks collecting specimens for the collection. So sometimes we do expeditions only to collect specimens. Sometimes we do expeditions to collect live fish and other animals for the exhibit. So when you go tonight, make sure you go see the Twilight Zone exhibit. A lot of the fish I'm going to show here are actually in the exhibit. So we went into the strip in April 2019 to collect specimens. And then 4 or 5 weeks later, in May 2019, we came back to collect live specimens for the exhibit, and this is what we found on the same reefs. So within a 5-week period, the water got so warm that the corals got stressed enough to expel the algae that was within their tissue. So that relationship that I said is very complex, it's very tight, it's very well regulated, and half a degree increase in temperature causes the coral to do this. They expel the algae, and because their tissue is transparent, their skeleton is white, so what gives them color is the algae that lives within the tissue. So if they expel the algae, they turn white, and if that lasts for long enough, because they get their nutrition from the algae, they starve to death, which is what happened there. Most of those white corals there, they actually died. If you look closely, though, there's little, the corals that bleached are almost of the same size, and then there's smaller colonies between the white ones that didn't bleach. So they were in a different cohort. They were products of different larvae, so they probably were more resistant to that temperature or more used to the temperature. They didn't die, only the large ones. Still, that's like a 50% loss in coral cover. They recovered relatively well because those little ones survived, but in other places, that's not the case. And as the climate gets warmer, those bleaching events, they get more, or they get bigger, they affect more coral, and they happen more often. So they used to happen once every 15, 20 years associated with a big El Niño. Now they happen almost every year because every summer the temperature threshold, the temperature is rising enough to cause these massive bleaching events. So what do I mean by deep reefs? If you think of like 99% of the coral reef literature, the scientific literature, every paper there is about coral reef ecology, coral reef evolution, almost all of that is based on science that is done by traditional scuba divers that did just the recreational scuba diving course and dive on that top 150 to 200 feet of water. So if you do an open water course in scuba diving, you can dive to about 60 to 80 feet. If you want to go deeper than that, you do an advanced open water course, and you can dive to about 130, 150 feet. And that's where most people stop. And most people stop there because to go deeper, the complexity of the diving changes, increases by, 5 or 6 order of magnitude, because you have to breathe a different gas, basically. So down to about 150 feet, all you do is you fill your tank with air, and you dive the same way you're used to seeing people diving, with a tank in their back, and then they inhale the air from the tank, and when they exhale, the bubbles come out. And then if you're a biologist and you want to study the deep sea, you use submarines. And if you have access to a submarine, they're so expensive, they're so hard to maintain, they're so hard to bring into the ocean, because you have to take them on a ship, and then there's the cost associated with the ship. So it's like tens of thousands of dollars to operate a submarine. And if you have access to that, you take them as deep as you can get them to. You don't spend any time between this area there between 200 and 500 feet. If you have a submarine, and the submarine can go to 3,000 feet, you take it to 3,000 feet. If it can go to 5,000 feet, you take it to 5,000 feet. So if you have a submarine, you usually go very deep. If you dive, you can only stay very shallow. So that whole area between 200 and 500 feet remained unknown, undiscovered, and unstudied, and still does. There's very few groups in the world that do it, because to do it the way we do it, by diving, you need a lot of training. And submarines, you can't really do it. Even if you were to get a submarine and you wanted to study that area with a submarine, in my particular case, I study fishes. It's very hard to study fishes on a reef with a submarine, because it's a highly structurally complex ecosystem, and fish hide. So you get close to a reef with a submarine, it's a big noisy, it's like a car size, big noisy thing with lights, and it looks like a spaceship probably to a fish. You get close to a reef, all the fish hide. So trying to study fish on a reef with a submarine is almost trying to study birds on a rainforest with a helicopter. It's not the right tool for the job. So in my particular case, to study that zone that we informally call the twilight zone, and every oceanographer is mad with me when I say it's the twilight zone, because there's an actual zone in the ocean that the oceanographers call the twilight zone that's much deeper than that. But that doesn't look like a twilight zone. I dove with a submarine to that zone, and it looks entirely dark. To measure light in it, you have to put an instrument. So it's darker than a night with no moon. So it's a new moon night, and you are at 600 feet or maybe 1,000 feet, which is still technically within the twilight zone. There's less light there than there is in a night with no moon out. So I don't like calling that the twilight zone. I call my zone the twilight zone because it feels like, and it's like twilight when you dive with it in it. Between 300, 400, 500 feet, you feel like you're diving during twilight hours. So to dive there, we use this equipment that's called a rebreather. It's much more complex than a scuba tank. It requires a lot more training. It requires a lot more preparation before the dive to use it. That's why most scientists don't want anything to do with it, because you almost need as much training to use that as you do to do the science. But we use it because it's a highly effective way to collect fish and to study fish, because unlike a submarine, you can get very close to the fish with it, and you can get to the depths we want to, and you can get there efficiently and effectively. So if you remember when I said to go to those depths, you need a different gas mix, it's because if you breathe air at those depths, air is mostly oxygen and nitrogen. So the atmosphere is 80% nitrogen, 20% oxygen. Those two gases, they happen to become toxic and narcotic if you breathe them under pressure deeper than 200 feet or so. So you can't breathe them. So we breathe helium instead, mixed with, yes, you can laugh about it. I mean, you hear it in a minute. You can only dive to those depths if you add helium to the mix. So we breathe a mix of very little oxygen, depending on the depth we go to, 5 to 10% oxygen, 10 to 15, 20% nitrogen, and then the rest of it, it can be as much as 80 to 85% helium. But helium is a very expensive gas. So if we were doing this without a rebreather, if we were doing this breathing off of normal tanks, every time you exhale at 400, 500 feet, it would cost you $10 for every breath. That's how much helium costs. So instead of doing that, we do a rebreather that recirculates the gas you breathe. So that's, I'm going to give you like the 10-second version of a rebreather course. So on a tank, when you exhale, the gas comes out into the environment as bubbles. On a rebreather, it doesn't come out. It goes back into the system. That's why it's called the rebreather. You breathe the same gas over and over again. So it goes back into the system. The system has a filter that removes the carbon dioxide. That's the byproduct of respiration. You don't want to be breathing that. It's like the same as breathing from a plastic bag. You don't, trust me, you don't want to be at 500 feet breathing from a plastic bag. So the filter removes the CO2. There's oxygen sensors that add the oxygen, only the oxygen that you need, and then you breathe that back. So what you don't see in this diagram there is helium. And that's because you breathe the same helium over and over again, making it orders of magnitude cheaper because you're not spending $10 a breath for 10 minutes. In one of those dives, you're just breathing the same helium over and over again. So it's on the long term, it's a very efficient way to do it. It just requires a lot of training, but we make it happen. When we finally got the training and we got to the right institutions, I've been trying to do that since I finished my PhD, which was a long time ago. And I only started actually doing it here at the Academy 11 years ago because I had the proper support from the dive safety office, and I had the proper support from the institution. So all of the stars aligned. We started doing this about 11 years ago. And one of the first questions we addressed was whether or not these deep reefs are a refuge. So up to 5-6 years ago or so, the whole literature that was very little that talked about those deep reefs, they assumed that they started their papers by saying that those deep reefs are very important because they're a refuge for shallow water reef organisms. Because of all of those threats that I explained, everybody thought that the deeper you went, the fewer threats there were. And then in order for this particular area to be a refuge, those two things there have to happen. So the species composition between deep and shallow reef have to be very similar. Otherwise, they can't be a refuge. And those deep reefs have to be far enough from those impacts to make a difference. So if there are impacts in both areas, then they're not a refuge. So one of the first things we started seeing when we started going to those depths was that that was not the case. The more we dove, the more we saw impacts of that same kinds of impacts that were affecting the shallow reefs. We saw them at the deep reefs. So we saw coral bleaching, we saw coral diseases, we saw coral damage by human activities, by anchors, by fishing lines, the same way we saw it in the shallows. The bleaching is slightly different though. Yes, temperatures are a lot colder at those depths, but corals, they're not about the absolute temperature. They are about how much that temperature varies. So in the shallow waters, corals in general are used to a 28. So for example, in French Polynesia, they're used to a 28 degrees Celsius temperature. That's about 82, 83 Fahrenheit. So if the temperature goes up by half a degree Celsius, if it goes to 28.5 or 29, they start bleaching. In those deep reefs, the corals are used to 18 Celsius. And if the temperature goes up to 18.5, 19, they start bleaching. So they do bleach the same way. It doesn't matter if the water is colder. It just matters that it is increasing, and it's increasing at the same rate as the shallow temperatures, as the shallow waters. What is different about those is that they're decoupled from the. I almost pulled the HDMI cable off. That would be worse than pressing the escape button here. So they are in a different water mass, but they are used to the temperature that's there year round. And that temperature in the different water masses, the ocean is highly stratified, they change in a different regime as the shallow ones. So when the shallow waters are warming, in general, the deeper ones don't. And when the deeper ones warm, the shallow ones don't. So when you see bleaching in the shallows, if you go and try to see it in the deep, you usually don't. And when you see it in the deep, you don't see it in the shallow. So because they were decoupled like that, and people only looked for bleaching even at the deep reefs, when there was bleaching in the shallow reefs, they never saw it, so they assumed there was no bleaching in the deep reefs. But the more we did it in different times, as we did in the shallow waters, the more we saw that there is a lot of bleaching on those deep reefs, almost as much as there are in shallow reefs. This is one of the deepest pictures I've ever taken. This is in the Philippines at about 147 meters depth. That's like 495 feet. This is one of those decorator sea urchins. It lives usually in seagrass, and it grabs anything that it can grab around it to use as camouflage. In this particular case, it grabbed that blue thing. There is a piece of plastic. So it grabbed a piece of plastic and it's hanging on to a fishing line. So that's in a reef that nobody has ever seen before. I am 100% sure of that. That's the first dive we did there. First time anybody ever saw that. And one of the first things I see is a sea urchin camouflaging itself with a piece of plastic, because it's the most common thing there is in its habitat, hanging onto a fishing line. So we have a paper coming out next month where we show that there's more plastic in more fishing lines and more fishing gear in general attached to those deep reefs than there is to shallow reefs, which was like mind-blowing to us, because you'd expect much more to be trapped in the shallows, because that's where, they're much closer to humans than the deep reefs. But for a number of different reasons, there's much more in the deep. Because, for example, there's less water movement. So the deeper you go, when you change into a different water mass, there's less influence from the wind. So the shallow waters, they get a lot of influence from the wind. So the waters in general move more. There's more circulation. There's more waves in the shallow water. So the plastics, they get removed from shallow waters more than they get removed from deeper water. So they accumulate more in the deep. And they might be a bigger problem for those deep reefs than for the shallow reefs. So this whole idea that there's fewer impacts in the deep reefs, the more we dive there, the more we realize there's actually at least the same, if not more, impacts in some cases than we'd see in the shallows. What about species composition? What about how those the species composition of those two habitats compares? So the other thing we did, in addition to documenting all of the impacts we could, was looking at fish communities between shallow and deep. We used that doing one of the most standard methods to analyzing fish communities there is. It's transect. So we just lay down a 20-meter long transect, and we count every fish towards each side, 1 meter of each side of that transect. And we do many, many times around the world. Horrible job trying to travel to all those beautiful tropical locations to count fish. Can't complain about that side of it. But we did it many, many times. We counted over 100 1,000 fish in almost 1,000 transects in 7 to 8 locations. And after doing many, many times, what we concluded was that not only those deep reefs were different, they were extremely different from the shallow reefs. So those numbers that you see there, that range from 85% to 99.5%, they're dissimilarity. So they're how dissimilar those deep communities are to the shallow communities. If you compare the shallowest reef to the depth gradient, the depth strata just after it, between 30 and 60 meters, it's 87 to 96% different. And then the deeper you go, the more different they get. So that whole idea that there was a lot of species composition similarity between deep and shallow was because when you look at the depth range of a species, that's usually what you get. And depth range is a very biased way of looking at this, because depth range is every record, almost every possible depth that species was collected before over all of history. So if we go to the collection, the fish collection right now, and we look at pick a species, any species, there's always a depth record associated to where that species was collected. And for some species, the depth range can be from 10 to 100 meters, in which case everybody assumed this was a shallow species that used the deep reef as a refuge. But it's entirely possible, and we saw that when we did the transects, that species was really only found between 60 and 80 meters. So it was a species that was very, very deep, and then once 100 years ago, it was collected at 10 meters depth. I don't know the reason. Maybe the water's too cold. Maybe it got chased by a shark and it ended up at 10 meters. So that is the depth range of that species, from 10 to 100 meters, but that has very little to do with the depth where that species actually lives. So when you do that comparison, you compare the depth strata using the transects, which is actually what species are there, the dissimilarity between those ecosystems grows a lot, as opposed to depth ranges. Now, something else that we did notice when we started doing this was that not only the communities are different, but those deep reefs are very different in terms of what is there, and a lot of what is there is new species. So these are just some of the new species that our team discovered and described over the past 10 years or so. I'm going to talk more about new species discovered towards the end of the talk, but I just wanted to put that slide there because they're all pretty. So I like pretty things. I like pretty species, and those are as pretty as you can get. And those are true new discoveries, by the way. I try to separate like a new description from new discoveries. A lot of the times when you go to a reef and you find a species, the fishermen will know that that species is there because they caught it, I don't know, 50 years ago or something like that. In this particular case, because they're so deep and most of these fish are very small, they're not caught by fishermen. So they are true new discoveries. When we go to a place that we caught a species that the fishermen knew about, we usually don't call it a new discovery. We just call it a new name or a new description or something like that. So I'm going to tell you a story about an expedition we did to St. Paul's Rocks in Brazil. This is an island that is about 1,000 kilometers from shore. So it's as remote as you can get, 1,000 kilometers from Brazil, 2,000 kilometers from Africa. So maybe almost halfway in the Atlantic between Brazil and Africa. I took that photo from the highest top in the highest peak in the island, which is about like 30 feet or so, 40 feet. Looking down into the research station. That research station is a station that is occupied by 4 people only. The Navy maintains that station only because if they don't maintain a permanent presence in the island, they can't claim the 200 miles of exclusive economic zone along the island. So they let scientists stay there for free. So scientists in Brazil use that a lot and love it. But it's very small. It can only be 4 people at a time. And if I were to turn around and take a picture looking the other way, it would be just open ocean. So I did that, and I turned around and I took a picture the other way, and that's what it is. That's the ship that took us there. So as I talked in the beginning, we need all of that diving infrastructure. We couldn't get that on that tiny research station. If you're lucky, you can dive there with a single tank if you take your compression and everything. We need much more than that. We need a lot of emergency gear. We need a decompression chamber. So we had all of that aboard the Alusha, which is the ship we took to that island. We did our usual surveys there. We did our transect surveys all the way down to 450, 480 feet. That's Hudson doing the transect there, and below him with the lights is Mauritius. So Hudson was a PhD student in my lab at the time. Now he's back in Brazil, but still a collaborator with us. We tend to collaborate a lot with people that go through the lab because the diving is so complicated to train that once you build the team, you can't really change it really quickly. So we just love collaborating a lot. Mauritius is our dive safety officer. He's with us every trip. He knows more about the rebreathers than anybody else I can think of. So it's very good to have someone like that on the team with you in the field, because if anything goes wrong, he knows exactly what to do. And he's also what we call an untasked diver. So those dives, they're so risky, there's so much going on that we have to have a person on the dive that is not doing anything. So if I have my head down collecting a fish, I want someone just looking over to see what is happening around. You figure out why in a minute. So let me see if this works. No escape, no escape. Okay. This is a video. I'm gonna click play here, and then I'll stop it. Oh, no, not that. There you go. So I'm gonna pause it there. That's 419 feet. That's Mauritius, he's filming. I'm down there. When he pulls back, you see. So those dives are very, very long. This is dive, just by the depth there, you see time. That's 15 minutes into the dive. But that basically means we just got there, we started working. It takes us about 10 to 12 minutes to go down to that depth. We go down as fast as we can, but we have to clear our ears and everything. And then at the beginning of the dive, we always stop, we make sure everything is okay, and then we stop a couple of times. So it takes us 10 to 12 minutes to get to depth. So we've been there for about 3 minutes, and we already have 83 minutes of decompression that we need to go to come back. So that's the bottom right number there, TTS. That's time to surface. So there's so much gas that gets pushed into our bodies at those depths that you can't go up very quickly. You have to go very, very slowly. And then you have to do decompression stops. So in this case, when we get to, let's see, if we stop the dive there, which we didn't, we just continued collecting for quite a while. If we stop the dive there, the first stop would have to be at 150 feet. And then from that depth on, every 10 feet, we'd have to stop. I'm getting this sign there. So all right, every 10 feet, we have to stop. And those stops got progressively longer. So the last stop in this case at 10 feet would be probably 30 minutes. And the total dive time would be, I don't know, 2 hours. We ended up doing a 4-hour dive that day, so we stayed another 10 minutes at that depth. And then we did our decompression. So I will let the rest of the film go here. Okay. So that's me down there with the light. I'm trying to catch a fish. You see the healing? That's the healing voice. So Mauritius is saying, look at the shark. So he's trying to get Hudson's attention. Hudson, look at the shark. So Hudson is a big guy, and the shark is enormous. So both, I'm going to pause it again here. Right there. Both Hudson and I, you can barely see it, I think. Both Hudson and I were so entertained trying to catch a fish that we never saw the shark. The only person that saw the shark was Mauritius, who was behind the camera. I don't know if you can see it or not. You see on the angle there, close to its pectoral fin, there's a trailing line there. That's a fishing line. So that kind of summarizes the whole idea that these deep reefs can be a refuge. This is a very rare species of shark to be seen by a diver, because it never, in the tropics, and this island is on the equator, it's 0 degrees in some minutes. In the tropics, it never comes up shallow because it likes the cold water. But I mean, we were so deep that we weren't in cold water there. So that's why the shark was there. It's a 6-gill shark. You never see it shallow in the tropics. You might see it shallow in colder waters, but it's very rarely seen by a diver. So this is the first time that this shark was seen by a diver in the whole area of the tropical Atlantic. And the first one we see is one that has a fishing line attached to it. So it's a very rare moment that when you see it, I mean, see that it's like signs of human presence right there. The reason we ignored the shark was because we were catching that fish. So we have an excuse, right? I mean, it's so amazingly colorful that we were completely hypnotized by it, and we ignored the shark. And so much so that we named it Tosonoides Aphrodite in honor of the Greek goddess of beauty, and that's how beautiful the fish was. But in the same dive, we saw all of that, all of that trash, all of that in the very same dive. So it's doing those dives is one of the biggest swings of emotions I have, is that we see this novelty, we see these fish that nobody has ever seen before, and then we turn around and we see trash in the same dive, human impacts. And in this particular case, it was really shocking because there was one building that island. It was that research station for 4 people. So we are a nasty, filthy species. So protecting those reefs is very hard because most of them are unprotected because people don't even know they're there. They don't know what species are there. In this particular case here, this is one of the best and biggest marine parks in Brazil. This is around Fernando de Noronha Island. And the boundary of the park is the blue line, and it completely excludes all of the deep reefs. So this particular example, it's in Brazil, but it repeats everywhere we go. Most places, the mesophotic reefs are outside the boundary of marine protected areas because people don't know they're there. People don't know they exist. So we try to change that a lot at the Academy. We have an exhibit there that I recommend you go see it. We try to put this on the media all the time. Every time we have a paper come out, we share it. We share the beautiful pictures. That whole shark video was shared by a whole bunch of news media when it came out first, along with the picture of the beautiful species. With taxonomy, we try to put funny names, not only the Aphrodite thing, but also lately we've been trying to use local names for species. So for example, the top middle one there, which is in the exhibit, by the way, this guy here, Silabrus frinifemina, is one of the most recent descriptions we did from the Maldives. And there's over 1,000 species of fish that were described and discovered by from the Maldives. Every single one of them was described by European naturalists. Every single one of them has a Latin or Greek name. This is the first of 1,000 that had a Maldivian name and was co-authored with a Maldivian scientist. So we try to do that everywhere we go. And a simple thing like that, in the Maldives, there's already a movement trying to protect more deep reefs because they know that species is from there. They know very few of them are protected, and they took ownership of it immediately because just by the simple fact that we put a name on it that was Maldivian. They got a lot of media attention because of the name, because of how pretty it was. And I'm getting waved the sign there of 0 minutes, and it was a beautiful thing. But it's causing change in the Maldives. So this is the exhibit you're going to see tonight. So when you see that sign, go in. A lot of the fish there we collected. So there's not many divers that go to those depths, so we have to collect our fish ourselves. So you see a lot of fish there. Most of them we collected ourselves and our own team. And this is the end of it. I think we'll leave questions to the end.
Thank you, Luis. We're going to hold questions till the end of the plenary session. I was going to say, you might have not seen that shark, but that shark sure looked like it saw you guys. So I was like, really curious. So it gives me great pleasure to introduce our next speaker, Dr. Anna Monfil, professor at the Central Michigan University and curator and director of the Central Michigan Herbarium. Also president of the Society of Herbarium Curators. Anna is doing service, a great deal of service to the Society for the Preservation of Natural History Collections, the Society of Herbarium Curators, and the Biodiversity Collections Network to promote specifically and advocate for the critical role of regional natural history collections to support research, education, and outreach. In the last 5 years, Anna has been part of Biodiversity Literacy and Undergraduate Education, BLUE, a network of education researchers, data scientists, and biodiversity scientists focused on creating inclusive educational materials that introduce the emerging workforce to biodiversity data and the extended specimen. In an effort to promote an inclusive biodiversity science workforce, On collaborators have worked most recently with the Smithsonian and the California Academy of Sciences to run workshops introducing students to opportunities and careers in biodiversity science. And her talk is Extending Biodiversity Collections, Engaging an Inclusive Biodiversity Science Community Through Research, Education, and Workforce Training. Please join me in welcoming Dr. Monfil.
You know, they didn't tell me about the do not press button. Must not apply. So first of all, I want to take a moment and really thank the planners for Spinach. I have been so impressed with Laura Eklund and Plenary and how helpful they have been. As I refer to her, my handler, Maricela, who has been the one coordinating this talk and getting the slides together. So I do hope that you know we appreciate the work that goes into some events like this. So what I'm going to talk about today is related more to the outreach, the education component, but to do that, I'm also going to bring in the research. And I firmly believe that we in the collections community have the potential to be the ones that move science into the 21st century because we have been the foundation of science to date. And so when you look at not just the specimens, they're very important, the data, but also the people, you guys, and the expertise behind each and every one of you, that's going to allow us to have the information that's going to move us forward in our science and also in the social aspects of science. So what I'm going to do today is walk through a little bit of some emerging issues or programs, use a research project that I do with the federally listed butterfly species to kind of highlight some of the process of trying to make extensible specimens. And then I'm going to talk about some initiatives that are both ongoing or finishing up or available. Okay, you'll probably see these documents a few times. I know there's going to be some talks that mention the extensible specimen or the digital extended specimen. There are two rather pivotal publications that came out about this that were derived from community consensus. These are based on the idea that was first put forth about the extended specimen in 2017 by Michael Webster. And it's the idea that around the collection and all the data is this constellation of information that informs research. So there's two groups, the Biodiversity Collections Network, which worked with the community to drive this document, looking for the future directions for extending biological specimens so that it moves from the digitization effort, which has been monumental and is still ongoing, to all the associated data that goes along with each and every specimen. Then the NASM report, which is the Biological Collections report, also came out about that time, was derived again by some of the same people as well as the community, and that again was about what are our next steps. And there have been a lot of projects moving forward. There has been a group working on an international perspective to make sure that this isn't just a US initiative done in isolation, but that we're thinking about this globally at the same time. There's also, and there's going to be talk about an action center, which has been in discussion. And then there's lots of efforts in terms of how are we creating the emerging workforce that is actually going to contribute to the digital extended specimen. So as part of the digital extended specimen idea, which is again, that specimen, but all that associated data and how we keep that interlocked together with all the metadata that truly informs that data, there's a couple initiatives. One is the holistic collecting idea. And this is coupled with the idea that we need to be strategic in how we collect. We need to look at what we have out there, think about this in terms of our collecting expeditions, but also when we collect, we need to be forward thinking. Coming back to the theme of how could this data be used in the future, or what other things can we collect along with the specimen, and how can we keep that all interrelated? And then that moves to the idea of the extended specimen of once we enter that into a collection and it's archived, how do we keep all those physical parts linked? And then how do we continue to link and inform the data in an exponential way? And if we're doing that and we do that correctly, we're going to be able to leverage our data in new ways and answer big, important scientific questions on a scale we haven't been able to do, even though many of us have been working doing this sort of manually. So this comes into the part that I'm going to talk about, which is more the social aspect of actualizing the digital extended specimen. And that's us. Those are the people making the collections. Those are people curating those collections. And it's the people doing the outreach to the stakeholders that are interested in this data. And so we may have heard of the idea of fair data, which is findable, accessible, interoperable, and reusable. And I would argue that the collection community has been a leader in this area and can remain a leader in this area because this is how we operate. We don't make people pay for what our data is, and we've been sharing it freely amongst ourselves and actually reaching out, trying to engage the larger, broader scientific community in leveraging this data and using it with all of its potential. But to move there, we actually need to make a pretty foundational change in science. And there is a lot of discussion right now about the idea of reintegrating biology. So moving from these siloed efforts in different subdisciplines to one where we think collaboratively from the start. The other one is to really think about inclusion and making accessible spaces. And that has a context on a global level. And we just heard a great example of that, of engaging local knowledge and local communities. It has to do with local communities within our own national borders and international borders, and creating the spaces that are more accessible. And that's the critical part. It's not just opening the door, but it's making the collections community a place people want to come. And so as you look at this open science, accessibility is a third of that. And that includes open data, but it also includes open access. And that's something that is difficult because we know when we want to make a publication open access, that's several thousand more. And that is just cost prohibitive. So what are we doing as a society to think about that and how the publications, the research that we do is truly available internationally? Then there's a concept of being reproducible. So is all that code out there? Are we sharing that widely? And then there's inclusion. And inclusion is a part, I think, that has to be there at the foundation of everything we do. Are we supporting diversity, equity, and are we creating spaces people feel they belong? Are we expanding our partners and our partnerships? And so that means when we look out and say, okay, a lot of us are trained systematists, and where and how are we reaching out to our closest neighbors, the ecologist, but what about the biomedical community? What about all those different aspects of science and making sure not just that we're saying, oh, come use our data, but that we're creating the type of data and the type of interface that allows that to be used by people that don't necessarily have our expertise, but would benefit from that. And then there's new pathways to science. We've heard of the term pipeline. Has anyone heard of the term pipeline? Okay. Well, that makes everybody get in the same pipe. And that is not inclusive. And now you hear about the idea of a watershed, that there's lots of knowledge, lots of people, different ways in, different ways out, and that we need that in science to keep our thinking creative and to also provide avenues for everybody to contribute. And so as I'm talking from moving forward, I'm keeping this open science idea in hand. The other thing is we realize that on a very local level, in my lab and with my collaborators, what are we doing ourselves to create extensible specimens? So we're not talking the vast technology and the great expertise it's going to take to integrate those. We're talking about, are we building it in a way that when that time comes and that tech matches up, that we also have the data in a way that's usable, and we have a culture that's embracing this open science component. So this is the Powashiek skipperling. It is a federally listed species in both the US and Canada. It has arguably only 2 remaining populations. They may no longer be or have any wild members. I think we saw 6 last year in the US in 1 population. And they saw several, about same numbers in Manitoba. So we've been watching this specimen or these species this species for several years since we noticed a very sharp drop-off. So they occur in tall grass prairies historically. When they occurred in the tall grass prairie, they grew the most abundant butterfly that was tall grass prairie specific. And now, this is what's happened in the last 20 years to the populations. So this has reached a critical component, and looking at conservation here is going to take a lot of people on a lot of levels working together. Since it's federally listed in both the US and Canada, we have to be having conversations. And we have to be working together, sharing data, sharing research, being collaborative, and practicing open science. So these are just a few of our partners. And I know that Spinach has been moving towards opening up, and I know there's going to be a series of talks from the Zumu people, if I said that right. And that's an example of where museums and natural history collections can work together, as they both have the collections. We work extensively with the zoos where they're captively rearing Powashiek. And we're also working with state and federal agencies, and we're dealing with 2 different countries. So this makes that data sharing component and that information sharing component a little bit more complex, but even more important. So they were both federally listed in 2014, or they were federally listed in both countries in 2014. And there is a drive and an interest in having the data drive our decision-making in terms of conservation and management efforts. And so to do that, there's these 2 advisory documents about recovery, but there is also a task that our lab has been working on, and that deals with doing an adaptive management approach. And adaptive management says, let's take the data, let's assess what we see, and let's make our management decisions based on the data and informed by the data. And there's a document and a process to this, but it's very much embedded in that idea of open science. Now, when we talk about data, there are lots of types of data that we have that we need to integrate to be able to ask questions. So we have historic Powashiek occurrences. This community really rallied to help us with that. We wanted to find every record of Powashiek in any of the collections. And I have to thank Chris Marshall, because I reached out to him. I said, okay, how would I go about finding out what people have in their collections specific to Powashiek skipperling? And at that point, LepNet had just been funded. And that had to be a really conscious choice for them to reroute their time and their effort towards just that one species. Because we all know digitization is something that's best done in volume. When you're working through it, you have a plan. Now you got to go pull specific specimens. Some of them may not have had any information on them. But what we did to sort of accommodate that time and effort is we had a joint publication that we did right out the bat where we published that data set. Now there's some mass data based on whoever was providing it, but what we were able to do is really expand our understanding of the distribution and historical distribution of Powashiek. We have ongoing Powashiek skipperling monitoring. So we have annual data with locations for the Powashiek as they're observed, because you can no longer collect this species. We also have in parallel the plant communities where the Powashiek occur. We have life history and biology work that's being done out of the zoos, population genetics, habitat management, ex situ conservation, which are these efforts in the zoos, as well as many other types of data that are only going to be valuable if we can aggregate them in a common space and access them relative to each other. And so that's where I have to bring in my graduate student, Sarah Hansen, and they have been working at trying to create at least a start to an idea. And I say that only because we only have access to the data we have access to. And that is, how are we pulling a data set together in a way and building a data set so when the time comes that that can link with national and international archives, that we have the data we need and the metadata that's attached to it. And when that comes together, when we look at associated taxa, when we look at management data and site history and environmental characteristics and geographical information, we've been able to start addressing questions such as, how have prairie fen plant communities changed over time? How have the communities where you find Powashiek changed over time? We can't tell you why they've gone into decline, but we can tell you what was different when they were abundant and what changed in the various areas over the time span and the populations that were there. We need to think about releasing some ex situ captively reared Powashiek. Do we just put them right back where they were, where it wasn't working out so well? Or do we try to use some of that historical data, which we have from natural history collections, to figure out what would be the ideal habitat, and then start testing some management practices to kind of manage that for the Powashiek? And all of that's embedded in the data, and all of that's embedded in the data coming from natural history collections. Now, another part of this is, we told you we published the data set, But a lot of our collaborators aren't really interested in getting the data in that form. And so this is a complex thing to do in terms of it can drive you crazy, but we get data, and we all know this, in what do we call them? Highly specialized data management systems. And they can vary by year by the same researcher. And we have to pull all of that in, but guess what? They want it back the way they gave it to us. So we can't just clean it up and say, look at the beauty of this. We have to now redigest it back so that they can do what they want to, what they know to do. And that's something to think about as we reach out to these communities, is what their needs are. What problem can we solve for them? Not, you guys have to do all the work to come to us, but what can we do to give you what you want in a reliable way that leverages all this expertise? And then the other thing that we've been able to do is do our best to define the ideal habitat characteristics. And again, that feeds in all these data as data layers. So this is the work that Sarah Hansen has been doing. It was part of a publication on the digital extended specimen, and it was just our example of trying to make an extensible specimen. And part of that is understanding all the people that touch the data as it moves along the system. And so this is our method, or this is our method after we learned how to do our method. So the publishable part. And it is, so we know we have a collaborative group. We need to all settle on how we're going to handle the data, this, lack of a better term, the spreadsheets we're going to use, the data we're going to gather, the delivery mechanism. And then we had Sarah, and Sarah was our data architect, where they built the database, and they built the data sets, and they built the on-ramps for our collaborators, so that when they went in the field, they could do what they're experts at, which is identifying the insects or the butterflies, and we could go back and then make sure that data could be imported. Then you have your field biologists. We were not the field biologists. We're not out there. Now we have been, but in the case of the Powashiek, that's not our expertise. I'm a plant biologist. But the Powashiek observers need to enter that data for us to be able to interpret it, and we need to have an agreement on how that's going to be facilitated. And I'll give you an example of when we go out and we do quadrats and we look at diversity in these systems, that it's a lot easier to walk a transect and throw a quadrat than it is to have random sites all over the fen. And so are you going to be hiking across 30 acres to get to all these different places, or can you kind of walk a north-south? And that's just an example of working with them to make sure that we're coming up with a sample design that meets our needs, but also is feasible given that these sites are not super easy to navigate. Then there's the herbarium curator, the person who's going to take in the specimens and integrate the specimens with the collection. And as you'll notice on this, we keep going back to that data architect, because it's the data architect in our lab, /Sarah, who keeps that all together and holds the final data set. So nobody, theoretically, is supposed to just grab what they have, produce a figure, go present it at a meeting. They go back always to the data architect to say, I need the most recent version of this. And that's really important. When you're down to 6 individuals, there's a big difference between I saw 12 and I saw 6. And depending on who's gathering the data and how it's being integrated, it's very easy to inadvertently send the wrong numbers out. So along with the fact that we're trying to do this, it became really obvious that we need to think about the education that's needed for people to be able to support this. And this is education that's going to need to come at every level. And I say that because we think of our stakeholders. That's the person consuming information that comes from natural history collections. That's also the people contributing to the data. And that's the people that might want to be data scientists. And so all of those people have to have enough familiarity with this process to support it and consume it correctly. And so you have K through 12. Now, I had a QR code on my very first slide. A lot of these things that I'm referencing are not necessarily my contribution, and one of them is this figure right here. So those are all cited, and you can actually find those through that QR code in a link tree, and I'll show it again at the end. But this is something that comes out of California, Berkeley, and that was done by Lisa White, which was reconfiguring the way we think of the process of science. So instead of our linear scientific method, which, by the way, largely ignores everything that we do. She has made us into this sort of feedback loop that takes into account and embeds within it the people doing the science, because that's critical, takes into account the value of observational science, and takes into account the data. So here it is to start thinking about how are we teaching people the way science is in practice, and how does it really reflect what we do? So then we have undergraduates who might come and take some classes in they may never become bio majors. They may be bio majors. A very, very small percentage of them may become data literate scientists, but all of them need to understand this data is there, it's available, and the fundamentals of how to look at this data. And so that started a project we have called BLUE, which is a biodiversity literacy in undergraduate education, and thinking about those core skills at the very, very intro level to provide an on-ramp. The upper level stuff, the stuff different scientists are going to be experts in, are all also important. So is genetic data, but understanding open access biodiversity ecology data and how you can use it or not use it is pretty fundamental to the way we practice science. So we've been developing, and this is also a module that Sarah developed, where they very simply have to come up with their own field sheet to record data, and then share with each other, and then learn maybe some of the things they could be thinking about, and things like data standards. And this is for all biology students to take, to start to think about that versus just handing them the spreadsheet to fill in. And, you know, the first thing they're going to say is, oh, this is a pain in the butt because everybody used a different method. Or guess what? Well, everyone wrote their dates different. And that is a real opportunity for them to think, wait a minute, there may be a data standard, so you don't even have to think about it. You just go use the data standards and get to what you think is of interest. So the other thing to think about is when we get into graduate education, how are we teaching them about best practices? And that includes methods, but are we focusing in on the methods that it takes to create open and fair data? And how much of how we educate our graduate students involves that? Are we talking to them about database and database design? They don't have to want to do that as their degree. I'm lucky to have found someone who does. But there are fundamentals of that that people need to understand in order to buy into it, which means support it. We're not going to make this happen if people don't do it at the very preliminary level. And then there's also, and this has been some work that BCon's been working on, okay, we have a data management plan, but what about a specimen management plan? And how are we developing that in a way that's really feasible for ecologists? And what are we doing with those specimens? And are we thinking about it from the day you enter a lab to do research? Are we introducing them to where their data is going to end up and to being part of this bigger open science movement? And then we have some great efforts that are out there to help retrain some of us. And that's the Carpentries, that's GBIF. But I think it's important to remember that we are vested in that. This very closely aligns with our research. But the vast majority of scientists out there do not take a Carpentries class and may not ever want to. But they are continuing to educate and hire people. And if we want them to prioritize data-savvy, data-centric science, we need on-ramps for those individuals that fit within where they are at their career stage and what their needs are of the data. And I showed this slide, and I will fully disclose, this came from my graduate student, Sarah, again. And what you see here is if you don't practice open science and you don't think about your data and you just get that publication done, you end up on the right. You got a publication done pretty quickly, you're going to be the superstar in your grad program because you're just churning out the publications. If, however, you join my lab, I'm making you walk that other route, which means you're producing incredibly important things. They are advancing this bigger collective, but our reward system isn't rewarding you for it. And so it's subtly and continually being undermined by the fact that it's going to take you longer to publish. Now, I argue now you're part of that data set, and so you'll have more publications long term. But in the short term, it's really hard to juxtapose those two methodologies. And I would also point out that we've all worked with, maybe ourselves, I'll fess up, been the one on the right. And you just had to get it done. And then today us really hates last year us as we try to figure out which final data set was actually the one we used in the publication so we can regenerate the figure that has to have some adaptation. So there is a lot of space for the person on the left and a lot of value there, but it's value you see downstream. And we have to start teaching that. The other thing is, if we are going to recruit people and we do want the data architects and the data scientists, we need to think about reward systems. We need to change a culture that's based in publications only. We need to make sure that this isn't treated like a service. I'm the real scientist. You just go handle all the data and the analysis and all the stuff that's in the background. So this is an important component as well. I think before we go into the future of collections, we also need to think about inclusion and accessibility. From the start, we can be leaders in this area. And we can think as we build this, how are we going to build this in a way that actually creates the community we want to be part of, the thing that makes Spinach such an awesome community. And that is opening our doors and providing these opportunities. So, of course, I have way more slides than I need, but this is sort of a slide introduction to Blue. One of the things that we've done with Blue is we've said, well, first we need to define what those core skills are, and let's reach out to the community and find out. So we did a survey, and out of the 7 main core skills, these are the ones that our community, and we had almost 400 people fill out the form, and only about 50% of them were introducing any of these concepts. But there was pretty unilateral agreement that these are the 4 things that we need to make sure that undergrads in any science discipline are understanding when they leave. So if we're going to make this and say they need to do this, we also need to create the open education resources that help people implement them. So here's an example of one we did, but this is an open education resource, and 2 things happen here. 1, I can cite the DOI number. 2, I can look at the number of people that have accessed it, which is 2.4 thousand, and then I can look at the people that have actually downloaded it. And if half of those people implemented something like this in the classroom, then that means thousands and thousands of students are being introduced to some of these concepts. Here's another one that we did. This was with a group that met through the digitization effort. And we don't want to forget that a critical part of understanding the data is understanding the methods by which we gather the specimens in the data. And so we produced this, which has been hit up 107,000 times. We also have to think of how are we creating these materials in a way that's inclusive. And this has been an effort we've done in collaboration with Rhea Ewing and Sarah Hansen to develop a set of modules that center the individuals and the scientists and are deliberate in including a diversity of individuals within the project. And this is something that's not often done, and it's as simple, and it's like here's one point that what they've done is this whole artwork aspect of it and storytelling aspect, but it does mean that we have to think consciously about the names that we're giving the individuals in our story. Where are we centering it? Is it always the US? Is it always Europe? And are we incorporating different ways of knowing? So what we need to do is we need to start presenting science as human engaged, and what does science and what do scientists look like? So I mentioned earlier, we ran a workshop yesterday with, I think we figured out about 60 attendees that was specifically geared towards bringing your identity to being a biodiversity scientist, how you do that, and how you operate in the biodiversity science spaces. We just did one that had over 100 participants at the Smithsonian, and we have several more coming in the next few years, which is really introducing students to the diversity of people and the diversity of jobs you can use in the biodiversity careers. Coming up is something Spinach is sponsoring, which is the Natural History Education Demo Camp. They have left it open for anyone who might have the start of a module or an interesting module that they think, oh, people might want to adopt this or work with it. And you can do this during this virtual experience. We have been doing this I think now 6 years, 5 years, 6 years, and it's a great opportunity to share your resources. The other thing coming up, anyone going to the mammal meetings, is that we've been working with MIPA, which is Museums and Emerging Pathogens, which is looking across the Americas, starting to think globally about disease and how that interrelates with what we're looking at in terms of collections and how we're engaging all the appropriate communities. And then last, and this is way too quick, and my apologies, the Biodiversity Collections Network is moving forward with BioFair, which is about building an integrated open fair data network. And a lot of that is also centered in the idea of thinking of the workforce, thinking of the communities and the stakeholders engaged. And I know there's going to be some talks coming up in this area as well, so look through the program where you'll see them. And then it really is unfair of me to claim any of this because almost everything I do is in collaboration with a lot of really brilliant and awesome people and a lot of opportunities that have been afforded to me through these initiatives. So there I go.
Great. Thank you, Dr. Monfils. So our last speaker in the plenary session is Dr. Julie Allen, an assistant professor at the University of Nevada, Reno, and curator of genomic resources and bioinformatics at the Natural History Museum. Her work covers bioinformatics, evolutionary biology, and public engagement, and is focused broadly on understanding the evolutionary, ecological, and biogeographic patterns that shape our world. And her talk is titled Integrating Public Participation with Automated Processes to Increase Specimen Digitization Rates. Please join me in welcoming Dr. Allen.
Okay, great. Thank you. I would also like to echo what Anna said in thanking the organizers and everybody for putting this awesome conference together, and in particular putting up with my lateness on everything, but I really do appreciate it, and for the invite in particular. Okay, so what I'd like to talk about today is a couple of processes that we're working on to really speed up rates of digitization. And over the last 10 years or so and more, there's been a massive effort in digitizing specimen data. In particular, getting that label data off of the labels into some sort of research-ready format. Now, around this process, there have been a number of different tools, pipelines, softwares, et cetera, that have been developed. And of course, this massive community that has also been developed around these processes, which is amazing because as new museums come in to start digitization efforts, they can always take advantage and jump on the groundwork that has been laid before. So this has been very exciting. So as part of the digitization process, of course, we're pulling data from the labels, and not only that, needing to get those labels into a georeference, right? So being able to actually pinpoint where that specimen was collected on a map. So these are the processes that I'm going to talk about today. So what is the status of digitization? I'll give you an example from one of the longest running TCNs. So there is currently a large gap between image specimen and actual data that we have, all right, and the digital data. So if we look at CERNAC, which is the Southeast Regional Network of Expertise and Collections, this is one of the longest running TCNs that we have. They have 4.9 million specimens that have been imaged and imported to data aggregators, iDigBio and GBIF, for example. 10 to 20% of those are missing collector and other textual data. 61 to 90%, depending on which collection you're looking at, is missing mappable coordinates, right? And this would be one of the collections that is most complete, right? Because it's been one of the longest running. So thinking about the newer TCNs that have started in the last few years, newer digitization efforts, projects, et cetera, you can imagine those numbers are actually a lot bigger. And this gap prevents us from being able to really use these data in important ways to understand where and when specimens have been in the past. So the project I'm going to talk about today is called DigiLeap. This is our NSF funded project. We're in the second year, or pardon me, we're in our last year of this project. It's called Leaping the Specimen Digitization Gap. Our leadership team includes UNR and University of Florida. We work with Notes from Nature, Zooniverse, and we have a number of other collaborators and important key members to this process. We have a really awesome group of advisors that we pulled into this project, and the idea here is that as we're building and developing these tools, we need to check in, right? Check in with the community, check in with people to make sure that what we're developing is actually useful, right? We're producing something that people want to use, is easy to use, etc. And throughout this process, we have a number of collaborative projects that have come on board since we started, right, including Big B, the terrestrial parasite tracker, et cetera, so, and many others. So what I'll talk about is a little bit of the ideas behind the project and the tools that we're developing. So the idea is to develop processes to leap that digitization gap. What we did was identify areas where we can speed up this process with automated processes. And in particular, what we want to do is leverage new AI processes, right? Machine learning algorithms, community science methods that we have going, and of course, the massive amount of effort that has already taken place, right? A number of specimens have already been digitized. So can we take advantage of the work that has already been done to really speed up this process? All right, so these are the three parts I'm going to talk about today. We're going to talk about automated specimen label digitization. Then I'll come back and talk about our georeferencing gazetteer, which I'll explain in a little bit. And then I'll talk a little bit about where we're going next. You know, what are the other kinds of data that we can pull from this process? This community knows more than any others the issue with trying to digitize label data, right? So, you know, label data have multiple different type fonts. They'll have sometimes handwriting, sometimes not, sometimes cursive, which nobody can read anymore. And so it becomes really problematic. And for this reason, museum specimen data in particular has been a little bit behind the times, right? In using automated methods effectively to be able to pull out that label data into some sort of research-ready format. Now, one of our first stabs at this was using a community engagement approach. So we've had Notes from Nature, which is an online platform run through Zooniverse, where people can come online. And what we do is we show them an image of a specimen, show them the label. We then ask them a series of questions to help type in that label format, label data. And this has been really, really fun. And what's really neat is up to this point now, we've been running Notes from Nature for almost 10 years. We've had 3 million specimens by over 10,000 volunteers transcribed through Notes from Nature. What's neat about this is that means 10,000 people have gone online, have been introduced to museum specimens, have explored or understand something about biodiversity, you know, been able to see some of these different collections, and are starting to understand a little bit more about how museums work and what museums do. I've been doing some experimentation, I'll say, on my students, my undergraduates, and asking them to help us do some Notes from Nature transcription, and then asking them before and after, what do they think about museums, natural history museums? What do they think about specimens? What do museums do? And their opinions do change after even just running through 10, 20 specimens on this. They start to have a better understanding that natural history museums aren't just about the exhibits in the front, right? But they're about housing all of these important critical specimens in the background. And they're also starting to understand the data that we need from these specimens. So this is a really fun and great approach. However, as you guys know, there are about 1 to 2 billion specimens in the United States. So at the rate we're going, we're going to need a lot more time. And it's the time we don't have, of course. So we're trying to think about how can we then leverage this massive community that we've built around digitizing the specimens, new automated approaches to really speed this up. So we're using a humans in the loop process. Through this digitization effort, we've identified 3 key areas by which we can build models. And the first is label segmentation, so finding the label on an image. The second, of course, is capturing the textual data. And the third is parsing that data, right? Finding the actual information that you need, date, specimen, locality, et cetera. And what we can do is, as we have this massive community of volunteers, is ask them to help us build the data sets we need to train models. So in each of these steps, we can ask volunteers to help build training data sets, help us validate the model, right? So we run a bunch of specimens through a model. They can say, yes, that worked, this didn't work, yes or no, right? And we can feed that back in to improve our models in the process. And then, of course, doing some error checking at the end of the day, right? So after we have our OCR label, we can have humans just go in and check and say, yep, it's good, go. Or make tiny changes that actually increase the quality of the data that's going back to museums. We all know museums don't have a lot of time, right, to actually spend checking, error correcting these labels. So ensuring that what we're handing back is high quality is really important. So what I'm going to do is just talk about two of these processes here, label segmentation and text capture. So what we did was create an expedition on Notes from Nature, where we put a bunch of specimens up and we just asked the volunteers, draw a box around all of the labels and then tell us what they are. Are they a barcode, right? Are they handwritten? Are they typewritten? So helping us to actually classify those images as well. We started with herbarium sheets because we have the vast majority of those in our data sets. And it's surprising all the different types of things that you will actually find on a specimen. You'll find labels, you'll find envelopes, right, with other specimens in them, et cetera, et cetera. So being able to build that training data set is critical. And then after that, we took all of the data from that, fed that into an object detection model, and then fed that back to the volunteers and asked them to tell us if it worked. So what we did was then say, okay, here's what the model found. This is a label. The model found this label and said it's typewritten, correct, yes or no, right? Did it miss any labels, et cetera. And with this process, then we can take that validation data from the volunteers and lead it back into training and testing, right, into that model improvements. As of right now, with label segmentation, our most recent process, we're at about 94% accuracy with those labels, which is great. And the last 6% really hinges on handwriting. It's always handwriting. Labels that have some typewritten, some handwriting, right? So we're starting to get into the really like nuanced, more difficult areas. But I think as we're labeling, like focusing in on what those issues are, we can really start kind of training even more specificity into those models. So I'll talk about a little bit about how we're running the OCR process, or optical character recognition. Those are just models that go through and read text, from an image. That's what I mean. So not all specimens are the same. Not all specimens should be treated the same or run through the same models. And even with herbarium data, with like the same kind of specimen, we're finding that very different models work differently for those specimens. So our approach is to take an image, take a label that needs to be OCR'd that you want it, and then run through a different series of pre-processing steps, OCR steps where you pull the text data, and then run that through a consensus approach. So we have all the data that's come from all of these different models, and then run that through a consensus approach, and then run some post-processing on that. So in that way, we kind of get the best of all worlds, right? If this model worked a little bit better with this specimen, this pre-processing method worked a little bit better with that specimen, et cetera. We have all of those going into the pipeline that we're developing. And the next step, of course, is to send this to the volunteers and ask, how did we do? Help us fix this. So they will then take this data. We show them an image of a specimen. We show them this picture of the text, right? And then they can go in and correct it, right? And again, that can then feed back into the model. So as of right now, this seems to be working really well, and we're pretty excited about it. We have an online service that's available for label segmentation and for this OCR process. And Rob Gralnick is going to be presenting this in a data camp tomorrow, and I can't remember what time, but please show up for that and he'll kind of walk through the process, but this is available for you guys to use now. And I'll also just say this has now been plugged into Symbiota as well. So making sure that the tools we're developing are actually linked to the pipelines and paths and networks that we use, right? So that if you guys are already working through a pipeline, you put all your stuff in Symbiota, that tool's there available for you already, all right? So we're now, like I said, we're on our last year of this grant. We're working on this data extraction piece, and that will be extended into these tools as we continue through that process. Okay. So the next step, I said, we were talking about the issue of getting data actually placed on a map. So getting coordinates on a map. So we've developed a service called BELS. This is called Biodiversity Enhanced Locator Service, Location Services. And the idea is that we want all of our specimens to be what's called georeferenced, right? And a best practice georeference is includes coordinates, so latitude and longitude, datum, so the type of system that's being used, and of course uncertainty, which often looks like a circle kind of around that point. So how certain we are that that point resides somewhere. This process can take a long time, right? You can imagine someone sitting down, looking at a location, and locations usually say something like this one here, which says, you know, granite outcrops, sagebrush, juniper, East Base Granite Mountain, Tooele County, Utah at 4,500 feet, right? So taking that information and then turning that into coordinates with an uncertainty is not an easy feat. Right? If you're really familiar with the area, it's easier. If you're less familiar with an area, it's much more difficult, right? And your uncertainty grows. And there are some amazing automated processes like geolocate to be able to put these coordinates on a map, which are getting better and better for certain areas, and for other areas in the world still have some work to do. So with that step, as each one, of course, needs to be checked and validated, it takes a long time, but fortunately there are now millions of georeferences that have been checked and verified over the last 10 years of doing this work. So our question here was, can we incorporate these into a global gazetteer in which localities can be checked against? You know, if you have a locality string that someone else has already gone through and painstakingly georeferenced, you don't want to do the same work, right? And we know that often specimens from the same expedition are often split and sent to multiple museums. So certainly many museums do in fact have the same localities on these specimens. So there's no need for us to have to repeat this work. Can we instead have a gazetteer that has all of those localities in it, so when someone goes to georeference, they can say, does this one already have a georeference? Great, give it to me, moving on, and then just have the remaining that need to be georeferenced in its place. Okay, so let's talk about the status of specimens in GBIF. All right, the vast majority, this dark blue line, this dark blue blob or purple blob, whatever color you're seeing there, is those with coordinates. The lighter blob are those that only have country data. And then of course, anything above that lighter blob are those that we don't have any coordinate data at all. The vast majority of these coordinates actually come from like eBird, right? Things where your phone is actually tagging the location with the latitude and longitude, and there's no uncertainty involved. So those ones are solid, right? And that's what the vast majority of them are. But if we start looking at, all right, let's just take a look at museum specimens, right? And ask, where are we in this process? So we have many with coordinates showing this darker area, but we see we have many more that have country only, and many more that are unknown on top of that. Okay. So if we actually look at the data within GBIF, what we downloaded all of the data and found there are about 1.6 billion specimens in GBIF when we pulled the data down. 1.5 billion of those are mappable. Some have some coordinates and uncertainty. And then we get all the way down to those that have a true valid georeference. We're at about 324 million. And that breaks down to about 39 million unique locations, all right? So unique locality strings, essentially, all right? So it's not many when you look at the grand scheme of things, but 39 million valid georeferences with localities could potentially do a lot of work, right? That can really help push us forward. So to build the gazetteer, we ingested all the unique locations, locality strings from all the data aggregators, well, all of them, two of them, GBIF and iDigBio, the largest ones. And all of the data that went into the into BELS, into our gazetteer, had to have coordinates, decimal data, and uncertainty. And of course, we gathered all the other information that we can, so we can, you know, provide back as much data as possible. So then we asked, okay, with this gazetteer that we just developed, how many georeferences can we add on to the data in GBIF that don't have them? So we had, as I said, 1.6 billion occurrences in GBIF. 32 million of those were georeferenced, and we were able to add 59 million to those specimens in GBIF. It's a drop in the bucket, right, when you look at 1.6 billion, but 59 million specimens that people don't have to go in and check and verify and validate is huge. And as our data kind of goes back in, we re-download GBIF every time, as our data goes back in and more people do more georeferences, we expect that, those leaps to be even bigger each time. Or as, newer collections come online, potentially a lot of their specimens will already have been georeferenced. So when we look at specimens, there are 227 million specimens. 27 million of those have been georeferenced, and Bells was able to add 16.6 million of those. So again, really a huge improvement from where we were before. All right. So just as a little validation, one of the questions we wanted to know is, are these data good? Can we trust the georeferences that we got? So this is a fun experiment that we did. This is a hand-curated set of adorable horseshoe bats where people went in and painstakingly identified the location for every single one with an uncertainty. And then we asked, okay, how many with those locality strings exist in Bells and how many match? All right, so we found 1,700 in the gazetteer of those total. And then we said how many either match with the coordinate or are at least within that uncertainty, right? The uncertainty of the hand-curated data set. And what we found was 78% overlapped or within that buffer, right? So the vast majority of these really are falling into an area that we can trust. All right, so this service is also online. This is the URL shown here on the bottom. You can just upload a file of one or many locations along with a specimen ID or whatever kind of ID you want, and then get the information back from Bells. And we've also now have this plugged into Geolocate. So if you go to Geolocate to automate your georeferences, you'll be given an option to first run all of your data through Bells and say what already exists before going through the automated georeferencing. Actually, you can do it in any order you want. You could do automated georeferencing and then check to see which one of those were in Bells and compare if you like. Right? So there's a lot of different options and ways you can do that, but they were really hoping that, adding this in particular to one of the most widely used tools in georeferencing is going to speed things up for a lot of people down the road. Okay. So finally, I just want to talk a little bit about the extended specimen, kind of what are we doing next? And, we've got this nice process of working with volunteers to build training data sets, and seeing where we're going. So I'm just going to briefly talk about projects that we're doing. This is in collaboration with Katja Seltman at the University of Santa Barbara on the Big B project. And one of the expeditions we've developed on Notes from Nature is measurements. So now what we do is have volunteers help us measure this space in between those two wings. That space is directly proportional to or related to the size of the bee. Right. So what we can do is have volunteers put that line there, compare that with the ruler, get those measurements and do a quick calculation. And we can now examine bee size changes over space and time, right, with museum specimens. Another part of the Big Bee project that we're doing is looking at hairiness of a bee. So the cute little bee on your left, hairiness has to do with how well pollen attaches to them, as you can see on the right, right? So that hairiness really can tell us a lot about their interactions with the world, right, and all of that. So we are working on developing models to actually pinpoint each pixel on the bee and say hairy, yes or no, high level, low level, etc. So we can then really compare that across the way. And then finally, this is a really fun project that we're working on now. This is with Dr. Nazi Balmaki, who's now at University of Texas Arlington, and Katja Seltman and Rob Grolnick, etc., where we are looking at all of the images of bees that have been taken over the last little while and asking volunteers to tell us, is pollen found on that bee, yes or no? Right? Because collecting pollen directly from a bee and identifying that pollen gives us a really nice interaction data set, right? In ways that you can't do before. Watching pollen, trying to get pollinator data, often people go and sit at a plant literally and watch it, right, for hours. And you might have one bee, and that's fantastic data, right? But it's really hard to get that at like a landscape level or over time level, right? So what we can do here instead is have all the volunteers look at all those images that have already been taken, say, is there pollen, yes or no, right? And then that can tell palynologists like Dr. Balmaki to go to those specimens, collect that pollen, and we're building machine learning models to identify that pollen as well. Palynology is one of the sciences that are fewer and fewer people are being trained in. So we think it's really critical that we get models built and developed to be able to identify pollen. And in this way, we can link actual pollen data to the actual specimen, right? And start looking at long-term trends and in particular declines in some of those interactions and relationships. So anyway, I hope you guys will think about it. Please feel free to use notes from nature, use our OCR services, bells, etc. And I'll be around. There are a number of people from our team will be here. Please come to the data camp tomorrow. And Rob will show you a little bit about the OCR services. And thank you.
Okay, before we move into, I think we have a little time for questions. Okay, so there are some mics in the middle of the room, and if you just come up and speak your question into the mic, and please direct it to a given speaker, and then the speaker can come up and address your question. And while people are getting warmed up, I have a question for Julia. And my question is, you mentioned machine learning to identify pollen. I'm interested in what you see as the future of AI for us for this process. I love and honor the fact that you keep humans in the loop in part in spite of technological advances, but could you please come up and elaborate? Yeah.
Yeah, it's a really good question, and we actually feel very strongly about keeping humans in the loop, and a big part of that is because as we're trying to solve some of these big scale data problems, like having the public be involved and part of this process is actually critical to start getting everyone kind of on board with what needs to be done, right, to move forward. So keeping humans in the loop is really critical. I think that in particular, machine learning is going to be transformational, in how we move forward. And in particular with palynology, when we're thinking about that, as I said, there are fewer and fewer palynologists. So for those sciences where we're really lacking some of those critical skills, it can be a game changer, but every single species and every single group is going to need a model built. Right, we're always going to need those massive training data sets, and maybe that's involved, maybe we start hooking up with iNaturalist to get those images, to start building those models, et cetera, et cetera. But I do think humans, you can't cut them out, and we'll be the better for it.
Are there any other questions? Yes, please step up to the mic.
Hi, my name's Sharon. I'm from the Field Museum. I have a question for Anna. Is she still around? Anna. Thank you for your presentation. It's really cool to start to see movement to teaching young people coming up through the system about the importance of all the things that we're trying to do. My question is, what are your thoughts on where they're going to end up? A lot of us are in museums, institutions with museums, and the lack of sustainability for those jobs that are doing the work that they're moving into, the pay differences. You talked about fair and equality and all of those things, but you get into the workplace, and where is the job that's going to pay your bills and keep you in the system?
I think that's a really good point. And I was having a discussion about this earlier, about the fact that we are not compensating, likely many people here, at their skill level. And so that's a little bit different of a question than the way I approach it, because people value what they want. I want this, or this is as we start showing the product of what we're doing, and we do bridge a lot of disciplines. So that if we're looking at careers that can utilize this data and these skills, they do move beyond what we're doing in the museum, but have the potential to feed back for valuing more of what's in the museum. That said, I think we need to be really realistic when recruiting people into this career about it. but also using the voices of those of us in the position of privilege of having those jobs, of advocating for the pay, putting it into our grants, and really confronting the idea that when we go to work and we undervalue ourselves, it's hard to continue to recruit people and expect them to value themselves. And I think definitely this next generation is seeing that and looking at our jobs and going, my gosh, you work all the time. And I'm an editor for a journal, and my daughter, who's mad now because I just mentioned her, and sitting over there says, oh, great, you get a raise in pay. I'm like, no. She's like, well, what do you do? I said, well, I read these papers, I do this. So then, what, you get more publications? And I'm like, no. And we are having a lot of trouble even getting on a lot of levels people to continue to volunteer their time. I think that valuing it comes a little, as much as we can promote that. We all confront the same issue of there isn't any money right now. So I don't know that that gave you an answer as much as I rambled about it, but I also don't want to delegitimize that point. If we're going to recruit people to this field, we need to be really honest about the compensation. But maybe we can leverage some of those skills we have to add value for communities that might have funds or might need the data that we have. I don't know. I'm open to anyone's suggestions.
Yeah,
I think that's
really
a
for the
real
whole room,
issue.
because there are lots of people here who are both at the bottom of the scale and at the top of the scale. And so for the people at the top to hear that, yes, we need all these people and they're going to do it all for free, but you still have to eat and you still have to pay bills, and everyone's like, give me more, give me more. So how do we get it up the chain to funders that when we say this is the pay for this job and these skills, that you don't come back with a, well, no, put them in a different department, pay them less. So thank you.
Yeah, and I think you're right.
So we do have to move on, but you can engage with our speakers and further discussion on these topics through Oxford abstracts and, of course, over lunch. But meanwhile, please join me in giving all three speakers, Luis, Anna, and Julia, a round of applause. And I'm going to pass it back to our co-chairs.
Great. Thank you all. So we have a few housekeeping things. While we have you all in one room, because tomorrow you'll be in 4 different rooms and all over the place. So as Shannon mentioned, please keep an eye on Oxford abstracts. I don't want to send any more emails to anyone, so we're going to put announcements there if things change. If things come up, you'll see those in Oxford abstracts. I recommend setting it to send you notifications immediately. It won't beep or anything, but otherwise you'll just hear about the icebreaker from me at the end of the day, and you want to hear about it before that. Speaking of the icebreaker, so tonight is the icebreaker over at Cal Academy. We will have 2 buses going from the Hilton. My goal for the night is to get you all there somehow, not necessarily on the bus. Those of you who need the buses, they will start loading at 5 o'clock. It's really easy, and a lot of you have already done it, to get to the Academy on public transportation here in San Francisco. So please use Muni if you can, but there are buses for people that will be going around and around. And the food tonight, speaking of food, I'm going to move on to food. I'm skipping our order. So, because this is near and dear to my heart, we do have folks in the room who have dietary restrictions, including myself. And you all filled out something when you registered. You probably don't remember what you said, but if you're a vegetarian, vegan, gluten-free, kosher, or halal, we have a special area of food in the Grand Ballroom B, which is right next door. So everybody else stay away from my lunch, and you'll be a much happier person for the rest of the week. I'm gluten-free, so don't touch my lunch. Everyone else that is gluten-free, vegan, all those things, come with me and we'll get our lunches together. The icebreaker. The icebreaker tonight, we've had a lot of people add the icebreaker last minute. We did order the food a couple of weeks ago. So it will be light bites. I'm giving you that warning ahead of time. There are lots of great restaurants near the Academy on 9th and Irving. Maybe plan a little bit of a light bite at the icebreaker and then hanging out with the new friends that you made today at some of the restaurants. So just a warning about that. I'm going to hand it off to Laura.
We'll try to get through this quickly because I think everyone's just getting hungrier and hungrier. So hopefully you all have your badges. If you don't, they are downstairs at the reception desk. Please go get them. As Mo mentioned, we have orange badges or orange banners for people who are involved in the conference. So we're happy to answer any questions. And our sponsors have the green banners. Please wear these at all times. The door monitors might actually be checking these going into rooms. This is for security and safety. But also just, it's a fun way to get to know one another. So I do have to confess that, San Francisco is kind of a big city. So I really recommend you take these off when you are outside the conference venue. You just don't want to advertise yourself as being from out of the area and maybe someone who's a little less attentive to their surroundings. So I'm very sorry to have to say that. You've all signed off on the code of conduct, the health and safety code, and we hope that this will not be necessary. But if anyone is feeling uncomfortable about a situation, or you feel like someone has breached the code, please do reach out to someone with an orange badge. We promise we will be both discreet and compassionate and do everything we can to take care of the situation. But again, hopefully that won't happen. For the health and safety code, that you've all signed, it talks about, making sure that you are feeling okay. If you are not feeling great, stay in your room. We are streaming these things live, and the talks will also be posted online pretty much right after they finish. So please take care of your own health, take care of the health of those around you, and just pay attention to that. And we're all going to have to watch a whole bunch of the talks online anyway, because we're going to have 4 concurrent sessions.
Okay, so we have a great week planned for you all, starting this afternoon with committee meetings. They're open to anybody. We encourage new people to attend committees. If there's a committee that you have a passion for, please join the committee meeting. Those are in the Franciscan rooms downstairs. We have the icebreaker tonight. The other big event that we want all of you to come to is the Friday annual business meeting brunch. It's important for the society to have people here at the business meeting to deal with society business. Plus there's some videos and it'll be awards and food. So you can add things on, just connect with the registration table for that. If you have any other things that come up, we do want to improve the way that the meeting happens next year or another time. If we can fix something now, please email SPNHC 2023 questions at Cal Academy. You've gotten a lot of emails from us on that email. You can also find that on the webpage. If you have any creative criticisms or critiques or suggestions, you can reach us there and we'll try to respond as soon as possible. But registration also can answer lots of questions. Have a great week.
Can I say one more thing?
She has one more thing to say.
A lot of you were in Chicago in 2019 when the Academy, how should I put it, got roped into being the host of this conference. And we were actually supposed to be the host in 2021. In 2021, well, we all know what happened there. So when we agreed to do this conference, hybrid was not a thing, didn't exist. You came to the conference or you didn't. And so I just have to put out a huge, huge, huge thanks to all the people who have managed to get this conference up and running in person and online. We have like 420 people on site this week, and we have over 100 more who are participating remotely. And needless to say, those are people who either can't get here for financial reasons, for legal reasons, because they need 10,000 visas to get into the United States. So many reasons that it's so great that we have this hybrid conference. It really did take a village to put this thing together, and I know it's not perfect, but huge thanks to our local organizing committee. A ton of people from the Academy, have devoted full-time, overtime, putting this together, trying to think of all of the details. We are not professional event planners. We do have a secret weapon. We have Ori Skyton Events, who are professional event planners, who have done a huge portion of this as well. So huge thanks to them. Huge thanks to all of our sponsors and our local organizing committee. That is too numerous for me to name. It's in the program. Check it out. Give them your thanks. And thank you all so much for coming. So please go enjoy lunch. It's right next door. That's where most of the meals will be throughout the week. And so you'll be spending a lot of time in there. Get to know one another. And thank you all for coming.